Welcome to Modern Group of Institution. Today in Pharmacology second BP503T, we study about bradykinin. So basically what is bradykinin? Bradykinin is a term derived from a Greek word. Brady means slow and kinin means to move. Because of this slow response, the name of the substance is given as bradykinin. Many of the trypsin and other snacks venom are acting on the plasma globulin and produce a small amount of substance which lower the blood pressure and slowly develop the constriction of the gut. Many of the substance like we know as a trypsin and some snack venom, they really act on plasma globulin, and this plasma globulin, when they act on this plasma globulin, they produce a substance which decrease the blood pressure, means contraction of the blood vessel, as well as developing slow contraction of the gut, that is the small intestine or large intestine. First of all, in 1960, a peptide contained 9 molecules of amino acid was isolated, and after that a decapeptide which contained 10 molecule of amino acid known as calidine was developed. So calidine and bradykinin, they both are known as plasma kinins, in which the bradykinin contains the 9 molecules of amino acid, and whereas calidine contains the 10 molecules of amino acid. Now, if we talk about the generation and the metabolism of the plasma kinins, this plasma kinin are polypeptide which are split off from plasma kininogen by the action of a specific enzyme kallikarins. The two important plasma kinins are calidine, that is decapeptide, and bradykinin, that is nonapeptide. As we studied in the previous size, nonapeptide means it contains 9 amino acid, and decapeptide, it contains the 10 amino acid. As you can see in the picture, the bradykinin contains 9 amino acid, that is arginine, proline, glycine, phenylalanine, serine, proline, phenylalanine, and arginine. Whereas the calidine, or it is also known as LYS, bradykinin, it contains one more amino acid, that is lysine plus bradykinin molecule. Now, what are kininogens? These two types of kininogens are present in the plasma. One is the low molecular weight kininogen, and another one is high molecular weight kininogen. The bradykinin is generated from high molecular weight kininogen by the action of plasma kallikrein, and the kinin and the kinin is developed from low molecular bradykinin is developed from the low molecular kininogen, whereas the another one, that is we known as calidine is developed from the high molecular weight kininogen. Kallikerins are mainly glycoproteins, enzymes which are produced by pre-kallikrein and present in plasma, kidney, pancreas, and intestine. Prekallikrein is activated by the Hageman factor. As we know, it is Hageman factor is the one of the factor which is present in the which is a part of coagulating factor, which itself activate the tissue injury. Kinin is also generated by trypsin proteolytic enzyme in snakes and various venoms. Now, first of all, Hageman factor, trypsin and kallikrein activate plasma prekallikrein, forms plasma kallikrein, and this plasma kallikrein in the presence of high molecular weight kininogen produce bradykinin, and low molecular weight kininogen produce calidine, and this calidine and bradykinin is metabolized by kininase first and second, and forms inactive fragments. Hageman factor, precalciferin, and the kininogens basically leak out from the vessel during inflammation or during any wound because of increased vascular permeability, which is caused by the histamine. Exposure of the negative charge surface promotes the interaction of Hageman factor with precalciferin. This activates the enzyme, and then bradykinin from its kininogen is formed. Now, how these both the kinins are metabolites. The both the kinins are metabolized by kininase enzyme. These are peptidases and having a half life of less than 15 seconds. Two plasma kininases were mainly responsible for the metabolism of kinin. First one is kininase 1. It synthesized in the liver. It is a carboxy peptidase that is released from carboxyl terminal arginine residue. And kinase second, present in the plasma and the endothelial cell throughout the body. It is identical to angiotensin converting enzyme. As you can see in the diagram, kinase 1 split of arginine, and whereas kinase 2 split of phenylalanine plus arginine from the bradykinins. As you can see in the picture, the calidine in the presence of aminopeptidase convert into the bradykinin, and this bradykinin with the help of kinase second convert into inactive fragments, and by with the help of kinase first carboxypeptidase M and N, it is converted into dextrorotatory arginine bradykinin and dextrorotatory arginine calidine. These both are inactive fragments of kinins, and these inactive fragments do not produce any therapeutic or pharmacological action. Now, if we talk about the kinins receptor, basically the bradykinin has two type of receptor. Both the receptor are GPCR in nature, having the similar type of effect. And further, if we differentiate between the two receptor, one receptor is B1 and another receptor is B2, and the function of B1 are basically they are very low level receptor present, but they are responsible for the inflammation and tissue damage and production of cytokinins such as interleukin first. They respond to the DES arginine bradykinin and DES arginine calidine, but not to bradykinin itself. Its metabolites are active on beta 1, whereas bradykinin is not effective on beta 1 or B1. These receptors are responsible for the inflammation, and hyperalgesia means excessive sensitivity toward the pain. Now, beta 2 receptor or B2 receptor of bradykinin is consecutively exposed in normal tissue. It selectively binds to bradykinin and calidine, and majority of effect are same. This receptor activated by phospholipase A and phospholipase C via interaction with distinct G protein. Whereas in B1 act by DES arginine 9 and bradykinin and DES arginine calidine. Their metabolite are activated in there are metabolite bind to B1, whereas bradykinin and calidine bind to B2 receptor. Now, as you can see in the diagram, when bradykinin metabolite bind to B1 receptor, it is a G protein type of receptor and GQ type of sub-receptor. It activates the phospholipase A2, which in turn produce prostaglandin from the arachidonic acid and leads to vasodilation and increased permeability. Whereas when bradykinin and calidine bind to B2, it Its subreceptor is phospholipase 2, which act on PIP2. This PIP2 follow the IP3DAJ pathway and calcium mobilization, which release and O generation and release. And in smooth muscle it causes contraction, and in vascular endothelium it's called vasodilation and increase permeability. Now, what are the various action of the kinin on various system? First of all, if we will talk about the cardiovascular system, these kinins are more potent vasodilator than acetylcholine and histamine. Their dilation is through release of NO and PGI2 generations. They markedly increase the capillary permeability due to separation of endothelial cells and cause inflammation. Injected IV quinine cause flushing, throbbing, headache, and fall in BP. Quinine have no direct effect on heart. Reflex stimulation occurs due to fall in BP. Now, if we talk about the smooth muscles, quinine induce contraction in the intestine, which is very slow, but they cause marked bronchoconstriction in guinea pig and asthmatic patient. Now, if we talk about the effect of kinins in the central nervous system, they produce potent pain producing agents potentiated by prostaglandin, stimulating nociceptive afferent nerves in the skin and viscera. In kidney, they increase the renal blood flow and facilitate the water and salt excretion by the tubule. What are their pathophysiological role? They are the mediators of inflammation. They produce inflammation, exudation, and leukocyte mobilization. Activation of B2 receptor on macrophages produce interleukin 1 and TNF alpha and other inflammatory mediators. They also produce pain by directly stimulating the nerve acting and increasing the prostaglandin synthesis at the area where inflammation started. Beta 2 antagonists block the acute pain produced by the bradykinin, but beta 1 receptor mediate pain for chronic inflammation. Another function, it is they produce hyperemia. Hyperemia means excessive secretion through the gland, regulation of microcirculation occurring through the local kinin production. Other roles of kinins, they produce dilation of fetal pulmonary artery, constriction of umbilical vessels, which is responsible for adjusting the fetal neonatal circulation. Their antagonist in the market available is icatibedent, that is B second generation beta 2 receptor antagonist. It is orally active, potent and selective, has a long duration of action greater than 60 minutes, and display high B2 receptor affinity in human and all other species. It is extensively used in animals to block endogenous and exogenous bradykinin, and humans to evaluate the kinins and inflammation, pain, and hypergeusia. The synthesis of kinin can be inhibited by kallikrein inhibited aprotinin. Actions of kinins mediated by prostaglandin generation can be blocked non-specifically with inhibitor of prostaglandin such as aspirin. Aspirin can be used as an antagonist. Sometimes the action of kinins can be enhanced by angiotensin converting enzyme inhibitor, which blocks the degradation of the peptides. Inhibition of the bradykinin metabolism by AC inhibit contribute significantly to the antihypertensive action. So this is all about the bradykinins. So kinins, as you in briefly, if we summarize, there are two types of plasma kinins present in our body. One is bradykinins and other one is calcitonin. And their metabolism produced by the carboxylase M and N, and they have various activities on cardiovascular system, nervous system, and other system. They are mainly responsible for the pain and inflammation. Thank you very much. That's all for now.